people want to hear what you're doing, and most likely you're being funded through taxpayer dollars, and it's how fun to kind of tell the taxpayers. What I like to say to people is, I work for you.
Hmm.
Science.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klonowski, your host for today's episode. Here's a thought for next Mother's Day. Thank your mom for all the antibodies. She truly sets you up for life.
Our guest today is Dr. Stephanie Langel, Assistant Professor in the Department of Pathology at Case Western Reserve University. Steph, welcome to the podcast.
Oh, thank you for having me. Feels good to be on and with you both.
Yes, exciting to have you. So, Stephanie, the first thing that we ask all of our guests is to tell us a little bit about their pathway to science.
Sure. Happy to share. It was a little different maybe than most, but I grew up very curious about nature. I grew up near a body of water and spent many summers as a young girl trying to catch turtles and frogs and snakes alongside this body of water I live near, and would ask my mom if I could get one of those giant baby pools and would fill it with as many animals as I could get my hands on. Had some, instances of snakes that bit me, and my mom is probably wondering, why don't I have a girl who's not getting bit by snakes? But it was a very adventurous childhood, and I very much thank my parents for allowing me to just explore. And that curiosity spilled over to, I went to a county fair when I was in middle school. My father had grown up with his cousins on their cattle farm, and my father always, I think, wished he was a farmer. He was a heating and cooling contractor fixing air conditioners and heaters, but I think always wished he was a farmer. And that kind of, I think, influenced me. And at that county fair, I remember thinking, I really want to do this. I want to raise livestock and show
Mhm.
animals, which was hard living in the suburbs-ish. Didn't really have a barn or a farm to do that. So my father, again, my parents just were very willing to try new things. And so I joined 4-H, which is an organization that allows you to be a part of many different projects that kind of culminates in showing at your county fair what you've done. But animals, raising livestock is the main project. And so I started with sheep, and my father built like a shed so that I could house sheep, and then we graduated to cows, and it was in 4-H I met farmers, and I started milking cows to make money for college, kind of as I was getting older and wanted to go to college, and thought, well, I'll try to make some money on the side. So I think my love of breast milk immunity and understanding how the lactating secretions of a mammal impact baby really started when I was on the farm, because I saw if a mom was sick, her baby likely was also not going to fare well if that colostrum was of low quality. And we had a way on the farm we could test that. And I I think I wanted to be a veterinarian for a while. I actually didn't know that you could be a scientist. I was a first generation. Parents had graduated high school, but didn't go to college. So I wasn't aware of the careers that were out there. So that trajectory, and I think having parents that allowed me to be an explorer, I think all of us as scientists, we kind of have that innate sense of like discovery, put me on the track.
That's really cool,
Yeah.
especially how it ties in with your current research.
Yeah, and I also like hearing from people that are scientists that don't have scientist parents, because I think a lot of people in the public might think that scientists beget scientists, but that's not always true.
Right. And I think the emphasis of having outreach programs for kids in areas of the country or different socioeconomic groups that maybe wouldn't know science as a career, I think investing in those programs is particularly important, because it is a way that we can, have science available to everyone. Yeah.
Absolutely. So let's move on to the fundamental question or questions that drives your research.
Sure.
So go ahead.
Oh,
Yeah, take it from the top.
okay. So it, the passive transfer of immunity from mom to baby is predominantly in the form of antibodies. IgG antibodies through the placenta, through breast milk could be IgG or IgA. And these are proteins that are produced from B cells that differentiate into plasma cells that make the antibody. I'm very interested in the fundamental dynamics of how that works and protects baby, but also mom while she's pregnant. I think, as I mentioned before, I'm just drawn to breast milk. I think there's less known about the passive transfer of breast milk antibodies and the mechanisms of protection. And really has led me to better understanding IgA as a molecule very rich in the mucosal surfaces. And the fundamental question is, how do I drive a protective breast milk immune response? And where are those cells that go to the lactating mammary gland? Where do they come from? What do they do? And then consequently, how does that prime them immediately, protect them in the immediate, but then long-term protection and development? Those are some of the fundamental questions we're interested in.
So, and just to make sure, since I am, as as Kim knows, I'm somewhat immunologically naive. But I think of IgG as like circulating in the blood, whereas IgA, which has a different structure, is found like secreted into mucosal surfaces. I usually think of it like in the airway.
Yes. So you're correct. Circulating IgG is predominant, although you'll find some IgA. But the majority of IgA, our body puts a lot of energy into generating grams of IgA, mostly in your gut, mostly excreted in your stool. The majority of IgA producing plasma cells are in the gut, but also in the airway as well, upper respiratory tract, really anywhere that you find mucus, anyway, secretions or just in that can be in contact with the external environment. There's a lot of IgA. And IgA structurally at the mucosal surface is different. It is, if you think of IgG as a Y-shaped protein, that's usually how we describe it. If I put my fingers up as a peace sign and then my hand is the bottom portion, IgA exists in the mucosal space as two of those Ys stuck together. And there's a special protein called a joining chain that can join those two. And it allows this molecule to pick up something important in the mucosal epithelium called a secretory component. This is like if there are, I just described two monomers stuck together with a J chain, the secretory component makes this molecule whole by protecting it from proteolytic degradation. It's actually for blocking of these enzymes that bacteria produce to try to cleave IgA. So it's a hardier protein and it's bigger than what's in blood.
Yeah, so that's the important thing that I think people don't realize is the IgA is sitting in the mucus, right? In the gut, right? Where all the bacteria and everything is, right? So it's all right together. So it has to protect itself.
Yes, and there is some really interesting work from Charlotte Ketzel's group. She's at Kentucky. She's now retired, but she had shown these beautiful pictures where IgA, the mucus and the epithelium, actually existed in different subsections of the intestine, and the IgA was below the bacteria so that it would protect the bacteria from going systemic in your gut. You ideally want to keep all of these bacteria at the surface and not penetrating the bacteria, which would cause sepsis. IgA is thought to and shown to protect those mucosal spaces and keeping this ecological niche of microbiota happy.
It's all one happy mix of host and microbes and
Yes, this kind of mutualism, which I think we're just starting to understand of just the diversity of IgA's functions. Not only what we study is antiviral functions. Can IgA block binding of a virus to an epithelial cell, but also how does it maintain bacteria in the space they should occupy?
in what ways does the mother's immune system protect an infant, and when does that start?
Yes. So if we're just thinking about passive transfer of antibodies, typically you will start to see that happening later in the second trimester, where it really peaks in the third trimester. This is why babies that are born preterm are particularly susceptible to disease, as they have not been able to receive all of mom's antibodies because that efficiency of transfer really peaks in the third trimester. And I think what mom has been exposed to or vaccinated against, those antibodies will remember. They will be specific to whatever mom has experienced. And that can be not only, again, pathogenic organisms, but also commensals. This idea that both placental but breast milk could help the microbiota establish itself in the newborn, which is really getting this onslaught of microbes in its environment. So antibodies really shape so much of the newborn, as well as I don't want to leave out my other immunological friends. Like there are cells in milk. Cytokine exposure during utero, IL-6 has been shown to be detrimental. Actually, you have increased levels of some of these inflammatory cytokines. So mom's immunity can make a difference, both in good ways and bad ways.
Yeah, I taught a new immunology class this semester, and we did a whole lecture on all of the things we're talking about today. And I will tell you that the students in that class were the most fascinated about this topic because we don't talk about it, right? Especially the fact that mom is like, as I mentioned in the intro, mom's setting you up for life, especially with your microbiome, right? And what your microbiome is early in life will predict how you will respond to infections, right? Or if you're going to develop autoimmune disease. So there's lots of things there. So can we talk about maybe some of what you think are the fundamental questions that have demonstrated the importance of maternal immunity?
Yes, and this is where coming from an agricultural background is helpful because farmers know, a farm kid's going to know right at the get-go if they're working on their farm how important colostrum is. And this is where it's really neat to think about the evolutionary divergence of how the placenta is formed between species. Because it is the case that a mammal that is able to transfer IgG through the placenta does not rely on breast milk to transfer IgG into the blood of the baby. However, cows, pigs, domestic animals, their placentas are too thick and they have no transfer of IgG during gestation.
Oh,
A baby piglet, a baby calf, without colostrum will succumb to infection because it has received no IgG through the placenta. So one of the key management things you have to have right on a farm is colostrum management and making sure that all of your newborn baby calves receive quality of colostrum from mom. The intestines of these animals uptake antibodies. They're very permeable during those first 24 to 72 hours. And so that key concept was really established when people started farming livestock and they understood this to be important. As we think with humans, it has been very true that I mentioned premature infants are more susceptible to disease, this being one of the reasons. And we have a lot of literature demonstrating that if you can boost mom's antibodies, it's associated with protection against that disease in the babies. So another kind of fundamental concept that placental transfer and breast milk transfer, although the mechanisms of protection are a little bit less defined, but are important in all species. But it is that the placental question, I think, is a fun one to talk about.
so it seems like most of the vaccines that we take are designed to elicit a good IgG response. So are they not so helpful for, say, breast milk, where I assume we're really looking at IgA?
Yes, it is true that breast milk does contain more IgA. And the trials investigating whether an intramuscular injection boosts immunity to mom and baby has really only looked at cord blood transfer and then associated with clinical protection. Less has been looked at for IgG and IgA. It is true that intramuscular immunization doesn't do as good a job at boosting IgA as does mucosal stimulation. And the SARS-CoV-2 really brought that to light, where you can look at breast milk from women who were infected with SARS-CoV-2 or vaccinated with an spike vaccine, and you see much more IgA in the women who were infected than vaccinated.
Interesting.
Now, it doesn't mean that IgG is useless. And actually, if you can drive a potently neutralizing IgG response and boost it in breast milk, you probably are going to
Mhm.
provide some protection. And that's what our lab's really interested in understanding is which one matters, IgG or IgA, both, one or the other, and then how do we drive a neutralizing antibody response to the mammary gland to provide this longer-term protection even as placental antibodies wane.
So, Steph, another thing that you study is something called maternal antibody interference. And so, what is that and how does it affect the infant?
Yeah. Yeah, this is it's a great question because it is the flip side that maternal antibodies are associated with protection because they can block infection of the pathogen. But when it has been demonstrated, when you have high levels of maternal antibodies and you try to vaccinate that neonate, the response in the baby is less efficacious than if the infant had lower levels of mom's antibodies circulating in its blood. So it can be considered a negative if the goal is to induce a robust vaccine-induced immune response on the baby. There are a couple of different ways we think this happens. If mom, the antibodies are circulating at high levels and the vaccine is based on either replication
Hmm.
or an antigen being presented to an antigen presenting, through an antigen presenting cell to a B cell, if an antibody has bound up that vaccine antigen, it can no longer present to your immune system, replicate in the cell. So there could be a direct blocking of the vaccine working. There are also really interesting receptors on B cells. There are inhibitory receptors. And it's thought that maternal antibodies could, on this stalk portion of the antibody I talk about when we make the Y, the long portion, it could be that that is engaging with these FC receptors and the B cells are not as able to mature and become plasma cells. So one of the things we're doing in the lab is to understand if you have this window of time when maternal antibodies are waning, but you need to give the baby a vaccine so it has time to induce its own antibody response. How early can we go back? What level of maternal antibodies matter? Or does the vaccine itself, can you kind of burst through this maternal antibody interference? So that's the flip side, the maternal antibodies that we're trying to understand.
I guess something we should also maybe point out is that these antibodies that go from mom to baby don't last forever, right? So
don't.
you have to vaccinate
Right.
the infant at some point because they will have a limited lifespan.
They do. They're right. There's a half-life. It's thought around 6 months that you really see
Oh,
the clearance.
that's longer than I thought.
I think it can depend on the antigen specificity, the subclass of IgG. We have
Mhm.
4.
Mhm.
But clinically, infants start receiving vaccines at 6 months.
They do.
And so that's generally thought to be a time. But I think for those of us interested in protecting, it is true. Infants in 0 to 6 months of age are the most likely to die from influenza in the pediatric population. They have the highest mortality rate. And that could be for a variety of reasons, but I think being able to vaccinate earlier in especially vulnerable populations like premature babies who don't receive maternal antibodies at the same level, we would want to know how do we manage that and how do we prove scientifically that it matters and we can protect these babies.
Okay, so that was great hearing about your research, Stephanie. But we also want to talk to you about your role as a host on the Immune Podcast. So, this is one of several podcasts produced by the Microbe TV Podcast Network. There's a network of podcasts, and probably one of the few focusing exclusively on the immune system. So, I guess the first question, the logical one, is how did you come to join the podcast?
Yes, I think this is a story that hopefully would tell students that it's okay to cold email and you never know where that may go because I was a PhD student and this series of podcasts really started with This Week in Virology.
Right.
Podcast started by Vincent Racket-Yellow, a virologist. I think there was a lot of us who were very engaged in that podcast from early on and liked what it was doing. And he didn't have an immunology podcast at the time. I was a PhD student, but I did like doing science communication. I remember emailing Vincent and saying, "Hey, you should have a immunology podcast." And he just said, "Well, why don't you start one?" And I said, "Well, I'm a PhD student. I don't know. And I don't really have the knowledge." He said, "Well, how about this? We'll try to get a couple other people. I'll do the production. Let's just see. Let's give it a try." And so we just started recording once a month. talking mostly about we dissect immunology papers and talk about current topics in immunology. The podcast has grown to 4 hosts: myself, Brian Barker, Cindy Leifer, and Vincent Racaniello. And I think our goal is to probably reach immunologists to keep them up to date on what's new. We also hope we get to reach maybe those who are interested in science, who may not be immunologists, who want to just be a well-rounded individual in their knowledge of what's going on in the scientific community. And it has been really, really enjoyable. I think I admire those who do the editing and the production because,
We do too.
yes, I am lucky that Vincent himself has been editing or hires somebody, I think mostly himself. But I prep for the podcast, I come on, we do the paper, but then I don't have to do any of the editing. So it has been only once a month, which is doable for me at my career stage. But I would encourage anyone, and like you all, what you are doing, I think, is really important. No matter what number of people we reach, I think just that we are putting something out there that has a scientific basis for the public and hoping we reach, those intents.
Yeah. So listening to one of the most recent episodes of Immune, I was really impressed with how well you guys kind of mesh. You generally get the feeling that you're not just colleagues but friends, and also that you enjoy the papers that you're reviewing or talking about or discussing.
Yeah. Yeah, we do. I think we've been doing it since, gosh, 2000 and I graduated in 2018. I think we started in 2016. So we had been doing it for some time. We've spent time together. We took a trip to visit AAI and interview people there. So I think we have a feel of each other, which I'm sure for you too as well. You learn about how to pass the baton back and forth. And I think because we are discussing people's work, we do treat the prep of the papers with that level of what if they're listening? I don't want to misinterpret. We know what they're saying, which is always a little scary, right? As somebody you're going on to discuss somebody else's work. But I'm glad you enjoyed it. Yes, it's been a fun endeavor. And I figure I'm reading papers anyways.
Exactly.
I might as well talk about them.
Exactly. Yeah, I mean, when you can do something and learn something at the same time, why not?
And probably you all have experienced this too. If I have to explain it over a microphone, I should make sure I understand exactly the way to make sure I get what I'm saying.
Exactly. And while our podcast, we're expanding out having guests like you, we really started doing mostly folks here at UGA. And even with that, I was learning about a lot of people's research that I had no idea existed on this campus. So just expanding your knowledge
And
in small ways.
it could have been a faculty member that you knew by sight and maybe you served on committees with or something.
Right.
And then you come ready to interview them on the podcast and you realize, I don't know anything about their research. It's,
Yeah, the other thing that I found fascinating, and that's why we always start our podcast, but this is how you got started in science, because that has really ranged across the board too. I think that's been one of my more interesting things to hear about.
Yeah. And I think it's good for people to know that there's not one cookie cutter way to become a scientist. And also just to know that we're all just, people trying to, you know, want to make sure we do our jobs well and we're there for our families. Like we're all, I think sometimes maybe scientists or especially when you're in the training stage, it can feel like, oh, are they really real people?
Right.
When it kind of breaks it down a little bit. Like, yeah, we're all trying to get through and.
you know, the, those of us who have podcasts are certainly trying to communicate, but I wonder what are the things that other scientists should be doing, can do, and should do?
So I really admire the younger generation of scientists who can use multiple forms of visual media to communicate a point. I think podcasts are excellent and they build a relationship with the host, especially if you're listening every week. But I think skills that I do not have, like putting a TikTok reel together to talk about B cell immunity to influenza viruses, these are things that younger generations know how to do. They're already on these platforms. I think we have to communicate in all realms
Mm-hmm.
of social media, but media in general, because our podcast, Immune, will only reach a fraction. The podcast here, social media has an opportunity to really expand who we reach, and particularly young people. But not only just young people, I mean, like you see these reels that come from across social media on Facebook where, many, like my mom, my parents are on, my dad. So I just would challenge and encourage younger generations. So if you're on those platforms, people want to hear what you're doing. And most likely you're being funded through taxpayer dollars. And it's how fun to kind of tell the taxpayers. What I like to say to people is, I work for you.
Mm-hmm.
I mean, my PhD was funded through the USDA to study an emerging porcine coronavirus. And I tell people, like, I'm here to share with you what I've learned because you paid for it. And
Right.
I want to be able to share that. So I think if we come at it with kind of a service mentality of serving the public, I think they feel then, the public, hopefully, feels valued and want to engage in this relationship, hopefully results in kind of continuing to value science as an endeavor.
Yeah, I think that's something that we need to invest in, whether it be organizations like AAI,
Yeah.
having a program. I mean, they have a policy fellows program, a communication fellows program would be great. Having other ways in which we're supporting students in those endeavors and being okay, I guess, if they're shooting a TikTok in our lab while
Yeah,
they're doing work.
I mean, right? I think I have a PhD student. She's really good with her reels and the visuals, and I encourage it. I mean, of course, biosafety we have to consider,
Right.
like, the type of work we do, but it's probably the case that if somebody goes viral on social media explaining something in a visually appealing way, they will reach way more people than I as an individual could. So maximize it. And not only that, like the way we communicate depends very much on cultural. And that's why having diversity in science, people in rural areas, suburban, urban, different socioeconomic levels. I mean, that's the only way I see us keeping science as a public good, is making sure our trainees represent all facets of the public.
Oh, definitely.
Yeah. Agree 100%. So, what is your favorite podcast to listen to? And it's okay if you say people, parasites, and plagues. But if you don't, that's okay too. It doesn't even have to be a science podcast.
I really enjoy Dr. Daniel Griffin's clinical updates every Friday through the microbe.tv. I think it is a nice way to stay up to date with things that the public is interested in. Particularly, people will ask me things that are clinically relevant about flu or infectious diseases, and Daniel Griffin's weekly podcast helps me answer those, because a lot of times people are not asking me about, BLIMP1 expression and B cells are maternal IgA and breast milk. So I think staying clinically oriented, if you're not an MD, has been helpful for me. But I also have a variety of kind of different things I dabble into. I will pop in to different fields even to try to understand, like, what's going on. There are, like, the journals also have podcasts. Like, sometimes I'll keep Nature has a podcast, JAMA has a podcast. So I try to stay well-rounded. Also, there's a couple political podcasts I tend to hop into.
It's hard to keep up because there's so many really interesting
Yep.
podcasts and there's only so much time.
I know. And of course, people,
Parasites
and
and plagues.
Yes. Triple P.
Triple P.
Sometimes the alliteration works against us.
Yes, it is true.
Now it's helpful to remember, though.
Okay, so our guest today is Dr. Stephanie Langel, faculty in the Department of Pathology at Case Western University. Stephanie, thanks for being with us.
Thank you for inviting me.
Links to additional information about Dr. Langel's research can be found on our Instagram account at PPP Podcast UGA. As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and a special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.